So, welcome to 11th standard biology. Now it is live. So, now we will see biology topic. I'm coming to here Windows, entire screen with student voice. Share. So, this is your session for the biology, chapter 2 of it's a Cambridge book. Cell structure. So, basically, how many types of cells are there? You know?
Two.
How many?
What types? No.
Two types. One is plant cell, another is animal cell. Okay, so we have to see their structure. Cell structure. What is the parts of the cell, and what is the functionality of this cell parts and all. Okay, so here cell types. These are the parts of the large sheet of paper. Okay, it is one activity he wants to do. Common cell structures. They're the common because there are two cells I said. Okay, in the two cells, see, one is a plant cell, another one is an animal cell. Basically, we have to discuss about these two, plant cell and animal cell. And here there is a bacteria is also there, have a common structures. Okay, so what are the common parts are there? That is important. And what is their functionality? We have to think. In the plant cell, what is the color of most of plants?
Green.
Green. Do you know the color of green will come due to one particular cell part? Okay, so we have to see that. We have to see that parts are very good, and even that there are vacuoles are also there. Vacuoles means where the food stores. Okay, so that is vacuole. In the animal cell there are small vacuoles are there, but in the plant cell there is a big size vacuoles will be there. And the CPU of cell is nucleus. Okay, so there is a core concept is that this is nucleus, and for this is also nucleus we have to think. Okay, so there are some major common parts are there, so we have to understand that major common parts. Okay. So cell membrane. Cell membrane, can you see cell membrane? Cell membrane is the inner part of the cell of the outer layer. So this is the entire cell structure. It's a piece of cell. Okay, this is a plant cell. This is animal cell. Animal cell. Only few things are common. Not all things are common. A few things are unique for the plant cell and unique for the animal cell. There are some common things are there, so we have to think about that. Okay, so here cell wall. There is no cell wall for animal cell. There is no cell wall for animal. The cell membrane is a common. Cell wall means the outer layer of this cell, and the inner side of the cell wall is cell membrane. So there is cell membrane, and for the animal cell there is only cell membrane. There is no cell wall. So these kind of things we have to discuss. So it is the first point is cell membrane. It is common in both. Cell membrane is common in both. So it is also known as plasma membrane, separates the interior of the cell from the outside environment. Okay, the plant cell is to outside environment. So after the plant cell there is environment, but after the animal cell there is another organ. So animal cells are inside the body, so that's why they don't need the cell wall. They need cell membrane, that is enough. Okay, so now cell membrane is separates the interior of the cell from the outside environment. Okay, so this is cell membrane. This is cell membrane, the interior. This is cell membrane. Okay. And next, cytoplasm. Cytoplasm consists of cytosol and eukaryotes and organelles. Cytosol is a gel-like substance made up of 80% of water, contains ions, salt, and organic molecules. You know, cytoplasm. Cytoplasm is, so this is jelly, a jelly type of thing inside the cell. So these are cytoplasm, and here also cytoplasm will exist here inside the animal cell also. There will be cytoplasm, cytoplast or cytoplasm will form. Okay, here he didn't mention that, but it is inside the cytoplasm. It is a jelly. It is not a dry piece. Everything is a liquid gel type thing will be formed. That is important. Next, DNA. Okay, DNA carries the hereditary information direct the cell activities and is passed from parents to offspring. Okay, so this is inside the nucleus. This DNA will you can get it. It is inside the nucleus, and DNA is the most important. DNA is the most important. It collects the information of the parent cell. Do you know when cell bifurcates? When cell bifurcates, it takes some previous information from the mother cell to child cell. So that is important. That is a it collects the information regarding the cell of the previous mother cell. Cytoplasm, cell membrane, and ribosome organelles responsible for the synthesis of proteins. Do you know when we take the protein food, it is to be break to become to get the energy. So the ribosomes will do such a kind of thing. Okay, so this is the entire digestion process or breaking the food cells, breaking the proteins. And classification of cells. So this is there are two fundamentally different types of cells are there. Organisms are classified according to cell type that which they are composed. Prokaryotes. Prokaryotes are composed of prokaryotic cells. They include bacteria, archaea. The prokaryote cells are usually unicellular or and are generally smaller and less complex than the eukaryotic cells. There is prokaryotic and another is eukaryotic. One is prokaryotic, another is eukaryotic. Eukaryotic means unicellular. 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 Okay. And the organelles of prokaryotic cells are not membrane bond. Not membrane bond. You can see this is a typical prokaryotic cell and eukaryotic cell. Okay. So this is prokaryotic. Prokaryotic. And this is eukaryotic. Cells. They are the two types. We'll see what is the difference between. So it is a unicellular. Eukaryotic are composed of eukaryotic cells. They include the protist, fungi, plants, and animals. Eukaryotic cell contains cell membrane bond organelles. Okay. So here there is no cell membrane. There is no cell membrane. So mostly it is just like bacteria. Okay. Bacteria kind of things are the examples for the prokaryotic. Eukaryotics are their cells with a cell membrane. The cells with a cell membrane. Okay. So this is the most important thing. The older classification of system of all organisms are divided into 5 ranks called kingdoms. Prokaryotic cells were placed in the kingdom Monera, and eukaryotic organisms were placed in the kingdom Protista, Plantae, fungi, and Animalia. These systems were based on morphology, appearance, and structure. So according to appearance and its structures, there is 5 types, but basically we will go with eukaryotic or prokaryotic. Okay, prokaryotic are unicellular without cell membrane. Eukaryotics are also unicellular but with membrane. It is there. Unicellular means only one nucleus is formed. Okay, is it clear?
Okay. Yeah.
Now, so it is kingdom. It is the bacteria, archaea, protista, and plantae, fungi, animalia. Okay, so they are the basic. Some definitions are here. We don't need to go such in depth. We have to understand that what is the kind of thing. So in 1970s, the use of DNA technically the discovery of two different types of prokaryotic cells. The discovery resulted to develop a system with three domains and six kingdoms. Three domains and six kingdoms. So it is a bifurcation of the types of cells. Domains are now the highest rank in the taxonomy instead of kingdom. Prokaryotics are divided into two domains, bacteria and archaea. All eukaryotic organisms placed in a third domain called eukarya. The four kingdoms within the eukaryotic domain remain the same: Protista and Plantae, fungi and Animalia. So we will come to this figure later. No problem. How the bifurcation is defined. It okay. And prokaryotics are eukaryotic without cell membrane. So these are cells without cell membrane. Cell membrane. Cell membrane. Prokaryotics are organisms that are made up of single cell unicellular bacteria, cyanobacteria, photosynthesis bacteria, archaea such as methanogens are examples of prokaryotics. Okay. Here there is no cell membrane, but rest all things will come. Chromosomes will come, and genophore, which is contained an irregularly shaped region called nucleoid. The nucleoid does not have a nuclear membrane. Even nucleus also does not have nuclear membrane. See, inside the cell, what happens? There is a nucleus. If you see, this is the nucleus. This is a nucleus. There is a nucleus membrane is also there. For nucleus is also its membrane is there. But in this prokaryotic, so there is no such a nucleus membrane is also. There is no, do not have a nucleus membrane. So this is prokaryotic without cell membrane, and without cell wall, without nucleus membrane and without cell membrane. Okay, so it is a basic prokaryotic bacteria cell. So it is bacteria. So its growth will be accordingly. So here the main points are capsule, cytoplasm. Cytoplasm is a common thing. Okay, flagellum, cell wall is here, and ribosomes, DNAs, nucleoid, cell membrane, capsule, each and everything will come. And if you go to bacteria, so he is explaining about the bacteria only. The prokaryotic chromosomes DNA is attached to the cell membrane by the region of chromosomes called origin. In addition to this chromosomal DNA, many prokaryotic cells have contain small rings, double-stranded DNA called plasmids. Plasmids. The cell membrane of prokaryotic cell is surrounded by outer cell wall. But prokaryotic, we did see that what is this? Previously, he mentioned that without cell membrane. Okay. Without cell membrane, cellular without cell membrane. We'll come back again. So the cell membrane of prokaryotic cells is surrounded by the outer cell wall. Many bacteria also have capsule outside the cell wall. The capsule protects the bacterial cell from the damage, dehydration, and engulfment by eukaryotic cells. It is also help the bacteria sticks to surface. Normally, he is explaining the bacteria behavior. Bacteria behavior. See, if without cell wall, the cell membrane of prokaryotic cells is surrounded by a cell wall. So if there is no cell wall, it will be dry up. Okay. So in this, can you see in the previous, it is a bacteria cell. So there is no strong cell membrane, but the cell wall protects. It is not strong. It is very light protector, and it protects the cell, the bacteria cell, uh, to get out of dehydration. Otherwise, it will be dry up and nothing will be formed. So this is about uh, prokaryotic bacteria type. Okay, and some prokaryotics can move around using tail-like structure called flagellum. Many prokaryotic have small hair-like projection called pili. So these are called pili. The small outer things are called pili. Okay, and these are the tails. These are the tails of the cell, and which is flagellum. Okay, and the inside the jelly-like thing is cytoplasm. So all these parts of the cell will come. Okay. Next, if I go a little bit for next, what is this? Okay, archaea. He is explaining everything about prokaryotic, only difference between bacteria and archaea. And eukaryotics. Okay. So now here animal cell and plant cell will come. So let's complete with prokaryotic only. And if we see a little bit go advanced here, eukaryotic, it is very clear. So the cell parts, how the cell behaves, each and everything will come. This is the main structure of uh, fungi, bacteria, and this is plant cell and animal cell. So that's about 2.1. Okay. If you come to here, bacteria. So I want to go with this definition again. Can you read up this first line, first para?
Organisms that are made up of a single cell, unicellular.
Mhm.
Bacteria, cyanobacteria,
Mhm.
photosynthetic bacteria, and archaea, such as methanogens, are examples of prokaryotes. Prokaryotic organisms can be found everywhere, even in extreme environments such as volcanoes.
Even in the extreme environment such as volcanoes, there its existence is possible. Okay. So, and most prokaryotic cells are small and therefore have a large surface area relative to their volume. Okay. So they're very small. This allows the cells to take and release the materials efficiently and replicate quickly. You learn about surface area to volume ratio in chapter 3. The structure of typical prokaryotic cell is shown. This is typical prokaryotic cell. And prokaryotic cells lack membrane bond organelles. Now I understood. Membrane bond organelles means, see, if this is a leaf, for example, you are considering a leaf. So what happens here? Everything is bond with cell membrane. Okay, everything will bond with, every small cell is bonded with the cell membrane. But this kind of bacteria, so it will not bond with cell membrane. Okay, so the cell is lives separately. The cell lives separately. There is no solidity of the organelle. Is it clear? Do you know what is the density of this organelle is very less. Okay, so their lack of membrane bond organelles means, so once this cell is completed, there is another cell will come. There is another cell will come. There is another cell will come. It is in general. There is a bond with cell membrane. But here, cell membrane is here, cell wall is here, everything is here, but it lives separately. So that's why, so these kind of pili will form and this kind of flagellum will form. It is a tail, and pili is some outer parts of the cell. Okay, and their cytoplasm contain a scattered ribosomes that are involved in the synthesis of protein. The genetic material of prokaryotic cells is usually a single cellular DNA chromosome called genophore, which is contained an irregularly shaped region called nucleoid. So there is no shape. Is there any shape? This is not a shape. Okay, so it is a nucleoid. Inside the nucleoid, there is a DNA. Okay, the DNA stores the information of the genetic information. Okay, that is a processing, central processing. So every information will be there only. Here, there is no a shape to the nucleoid, and the bacteria cell lives alone and with some pili and even cell wall and cell membrane. They are the only protection for this cell. If there is no these two things, then it will be dry up and it will be dead. Okay. And nucleoid does not have a nuclear membrane. So there is no membrane. If there is a membrane, so then you will get a structure for the nucleus. But there is no nuclear membrane. So it is just like everything is just like a jelly shape only. Okay. Even so these are small bubbles are there, na? We have its name. We have its name. These are ribosomes. How the ribosomes are look like? What is this? The ribosomes are look like small bubbles. Remember it. In the cell, in the plant cell, animal cell, these will come. They are the ribosomes. And this is plasmid. So this is plasmid. And it is a cytoplasm. It is a jelly. It is a jelly-like thing will come. It is a cytoplasm. Okay. And this is a DNA. There is no nucleoid membrane or nucleus membrane. Of course, we have cell membrane and cell wall, but the bacteria is not cell membrane bonded organelle. It is not cell membrane bonded organelle means it lives alone. It lives alone. Means huge number of these kind of bacteria lives together, but each and every one is separated by single. But in the general, the organelles are not separated like this. They're connected with the cell membrane. They bonded strongly with the cell membrane. So immediately after one cell, there is another cell. Okay, and cytoplasm has come, cell wall, and this is mesosome. It will come. It is mesosome. Is this cell parts are clear?
Yeah.
Okay. Nucleoid is in the cell. There is a nucleus. It is a core center. It is a core information provider. Everything will be planned there only in the nucleus. Okay. And without nucleus, nothing will be decided for the bifurcation or for the whatever the synthesis of proteins and even each and everything regarding the cell functionality. Okay. And this flagellum is nothing but the tail-like parts are called flagellum. And next, bacteria. So can you read above this first paragraph?
Most prokaryotes in the domain bacteria are microscopic, single-celled organisms. Fossil evidence dated to between 3.7 and 4.3 billion years old confirms that bacteria were the first type of living organism on Earth. Today, they are still the most numerous type of organism in the biosphere.
Wait. So it is single-celled organisms. Okay. He given the general structures of prokaryotic bacteria cell. So he is giving one bacteria cell because prokaryotic. He given the bacteria example, but there are several. Other examples. So most prokaryotic domain bacteria are microscopic single-celled organisms. Single-celled means only single cell lives. The functionality it is autonomous. It is the autonomous decision maker of the cell. Means all cells with the similar, but the functionality, each and everything related to this only part. It is single-celled organism. Okay, there are number of such a single-celled organisms will form. The bacteria count will be increased. Bacteria count will be increased. And fossil evidence dated that 3.7 and 4.3 billion years old confirms that bacteria were first type of living organisms on the earth. When earth formed, the temperature cooled down, the water developed, water formed. After that water, this kind of bacteria is formed. First of all, bacteria is formed, then bacteria changes and each and everything changes. The number of modification has come in the environment. So bacteria have very diverse metabolic system, making them extremely adaptable. They can survive almost every environment on the earth. Bacteria are common in most low salt environments of moderate temperature where sunlight or organic compounds are plentiful, and inside are plants and animals. Okay. So it can survive almost any environment. Bacteria need a little oxygen to survive because they have many ways of extracting energy and fixing carbon. Okay. Bacteria can obtain energy from the sunlight. There is a photosynthesis, or by reducing inorganic compounds such as sulfides or ferrous ions or chemosynthesis. Okay. So it is a chemosynthesis, there is a photosynthesis. Photosynthesis is, what is the photosynthesis? It is developing the food from the solar power, from the sunlight. So bacteria plays an important role in the ecosystem because they break down many kinds of substances, including plants and animal remains and waste. We see, I think we have seen in that ecosystem. I explained in the ecosystem what the process of what is the major requirements of an organelle. Do you remember this? There are five major requirements to exist in the ecosystem. For any organelle, so they need five kind of things. One is energy. I remember one is energy is needed. Next, tissues, reproduction. Okay, energy, reproduction, tissues. There are two more words are there. There are five things. Okay, I will recall that. We have to look at that chapter in the fourth volume. Okay, how the ecosystem, for every ecosystem, the five things are main things. One is energy, reproduce, that is reproduce, that is reproduction, and tissues. There are two more things are there. I'm not getting right now, but I will recall it by next class. Okay, we have five main things in order to sustain in the ecosystem. So next, gram-positive and gram-negative bacteria. Can you read this?
Bacteria have mesh-like cell walls that are made up of polymer called peptidoglycan, also known as murine. Different species of bacteria have different cell wall characteristics based on the structure of their cell walls. Different species of bacteria can be classified as gram-positive or gram-negative using a technique called gram staining.
Okay, so using the technique of gram staining, and see, bacteria have mesh-like cell walls. Mesh-like cell walls means, you can explain, the cell walls like a behavior. It is, we can't specifically clearly mention about that. So that is a mesh. Okay, and made up of a polymer. Like polymer is called peptidoglycan, also called murine. Different species of bacteria have different cell wall characteristics. See, have you ever seen normal cloth? So if you see a normal cloth, you can, that is generally. But if you see this cloth, there is a furnace there. Okay, so depending upon the cell wall also, this bacteria's outer layer could look like differently. That will depend upon the cell wall, cell wall or cell membrane. Okay, depending upon the cell walls or cell membranes made up of polymer. Polymer means, I said that it is not only a single cell will form. Okay, there are, for just such a unicellular things, it's a infinitely many such as unicellular things will form. All are separated because they are not membrane bonded cells. So the organelle look like no shape. The organelle look like no shape. So these kind of things will form by this prokaryotic cells. Prokaryotic cells, because they are not bonded with the cell membrane. And this infinitely many such a prokaryotic cells, we can see it look like it's a made up of a polymer. Polymer means infinitely many things will stick together. Okay, different species of bacteria have different cell wall characteristics. According to bacteria, bacteria is a lightweight. Bacteria we need, but there is a positive bacteria associated, there's a good bacteria, there's a bad bacteria. Okay, sometimes good bacteria will help us even making a curd. Okay? So that is, what is that? What we call it as? There is someone, not getting that name, but uh, there are good bacteria. There are good bacteria and bad bacteria also. We need bacteria. Okay, depend upon the cell walls and structure of the cell walls, we can define different species of bacteria is there and classified as gram positive or gram negative using the technique of called gram staining. Gram staining. Gram staining involves adding a purple dye called crystal violet or bacterial cells. Okay, so it is become a purple color things will come. It is a bacteria. It's a huge bacteria. Okay, so in this bacteria, can you see bacteria, gram positive bacteria are stained purple and the gram negative bacteria stained pink. There is a pink bacteria are there, there's a purple bacteria are there. This is a light micrograph was producing the light microscope. Even microscopes are also 2 to 3 times. We have to understand that. Okay. So here, depend on the color we can define one is the gram positive, which is blue. Gram positive is in the blue and gram negative is in the color of pink. So this is how we separate them. Okay. So can you read from here onwards?
Gram-positive bacteria have a thicker layer of peptidoglycan that absorbs and holds the stain, so they give a purple or positive result. Gram-negative bacteria have a much thinner layer of peptidoglycan that does not retain the stain as well, so they give a pink or a negative result.
Okay. So this peptidoglycan means it is a polymer. It is a cell wall of the outer layer. It's a polymer of cell walls. It's a polymer of cell walls. Means when cell is here, the outer portion will be the cell wall only. Have you ever seen? Do you know fishing?
Yeah.
I will see in the commercial fishing dock. You can see uh, the boat collects the fish from the sea with the net. So if you open the net, everything will be collapsed. Their number of fishes. So every small fish look like this this particular bacteria, and collectively having these kind of bacteria is called peptidoglycan. And depending on the color of peptidoglycan, we can define one is gram positive and another one is gram negative. Okay, Gram positive is with the blue color, gram negative is with the pink color. Pink color. So hold the stain so that they give a purple, or it is marked, it is colored. Okay. The gram negative bacteria have a much thinner layer of peptidoglycan, and that does not have retained the stain as well. So they give pink or negative result. Okay. So if you go further, so it is, there are numerous types of gram negative, gram positive bacteria. For example, gram positive are cocci, or spherical bacteria that include Staphylococcus and Streptococcus, which can cause serious disease or death in human. An example of gram-negative bacterium, it is a gram-negative. It is a gram-negative only. Okay. Again, an example of gram-negative bacterium is called cyanobacterium. Cyanobacterium were once called blue-green algae. Blue-green algae because they contain chlorophyll. Whenever green comes, then chlorophyll will come. Okay. Now we know how cyanobacteria are actually prokaryotes, and so they are placed in the bacteria domain. The cyanobacteria often form a dense colonies in shallow estuaries or freshwater. Some species form a large colonies known as blooms, which produce toxins. These toxin blooms can kill fish or other aquatic life, cause illness in humans. So gram negative or dangerous. Gram negative or dangerous bacteria. So there are sometimes these can kill the human also. Okay. So this is one example he given. What is this example? The Staphylococcus and Streptococcus, which can cause serious disease or death in humans. Okay, he explained it breaks the system. It impacts the nerve system. It impacts the brain nerve system sometimes. Okay, so and they can't easily. It is a scanning electron microscope. With the help of scanning electron microscope only we can see. So it is cocci. Okay, and next one more example he given, blue-green algae. So it is also toxic. So can you see what is this? What is
Algae?
algae? Okay, and the collected old water, it will be developed. This is algae. Okay, so there's a even. Do you know mushrooms? Do you know mushrooms?
Yeah.
Mushrooms also, we have, there are some positive mushrooms and negative mushrooms. Some mushrooms are good for the health, some mushrooms are very bad. They are poisonous. So here also some poisonous algae is formed. Poisonous algae will form. So those kind of things are come from the domain of gram-negative bacterium. Okay, is it clear? Regarding gram-negative, it is not good for human. Okay, and we'll go to the next thing. Archaea, prokaryotics in the domain. See, here we learned about bacteria, and next one is archaea. The prokaryotes in the domain archaea include extremophiles. These are organisms that can live in extreme conditions. So those who live in the extreme condition, so they are called archaea. And areas of high temperatures, they live in extreme conditions, high temperature, low temperature, upper atmosphere, alkaline environment, acidic environment, salty environment, environments with little or no oxygen, areas without light, petroleum deposits in the deep underground of the earth. all these extreme climates, the archaea will form. Archaea will sustain. Their ecosystem will be developed there. Okay. So even, do you know what is this lipids? What is mean by lipid? What are the lipids? Do you know?
No.
What is lipid? What is lipid? Fats. One can call it as fats. Fats.
Okay.
They are the lipids. Next, the lipids. It's a fat. Okay. So archaea hold records for living in a hotter place, 121 degrees, and the most acidic. Can you read up point by point?
Archaea holds records for living in the hottest places, the most acidic environments, and the saltiest water.
Mm-hmm. What is the pH value?
However,
Do you know pH?
of water.
Mm-hmm. No. pH value.
Okay. Like the acidity of something. 0 to 9.
14. What is the pH value of water?
7.
7. So here we call 0 to 7 are base, and these are called acids. 7 to 14 are acidic in nature. 0 to 7 are basic in nature. And water's pH value is 7. And whenever it goes to increase, the pH value will be increased. Okay. So here, mhm,
Is it less than 7 acidic?
I think it is less than 7 is acidic. Less than 7 is acidic, and 0 to 14 is basic or base.
Yeah.
Okay. So the most acidic environment where pH is 0, still it will be sustained. The saltiate water, about 30% of salt, it will sustain. However, some archaea will live in less extreme environments such as open sea. There are many different types of extremophiles. The alternative name is extremophile, means any environment they will live. They will live in any environment. And hyperthermophiles such as Pyrococcus furiosus can survive in very hot environment up to 100 degrees Celsius. They can also withstand extremely high pressure. Sulfilobus is genus of archaea that live in a volcanic spring. Sulfilobus or thermophiles as well as acidophiles because they can survive in both high temperature and high acidity. So is a special kind of things which live in extreme situations. Next, although archaea and bacteria are now known to be very different organisms, they both are different. So for the bacteria, it is not possible. For the bacteria, the temperature should be normal. Then only their growth will be possible. Archaea is different. It is totally in the extreme situations it is possible. Okay, so both are different organisms. Both are different organisms did not recognize these groups as distinct for a long time. Many species of archaea and bacteria look very similar. The extreme habitats that archaea occupy also make them difficult to find and culture in a laboratory. Okay, so please can you read up this third para?
The ability of archaea to survive in extreme environments is due in part to their unique cell membranes, like other living organisms. Archaea possess a cell membrane composed mainly of lipids. Cell membranes need to be fluid to enable cells to rapidly respond to external conditions and allow proteins to easily move in and out of cells. You will learn more about the structure of cell membranes in section 2.3.
So that's why you see these are also lives in the lipids. Lipids means it is the fats. In the fats, it is not easy to enter the cell to and the leaving the cell. See, if there is a cell, so the some things should become inside and some things should go outside in the cell. So in order to get the energy or synthesize process, so the cell has to allow some things, some energy particles. So once they enter, they will synthesize, and after synthesize, that becomes the energy. Okay. So this is normal in the general cells, but in the lipids, what happens? They will not allow easily. They will not release easily. So they need a special special thing to do each and everything. So their growth is they will not move rapidly. Okay. So once, do you know, in last 2 years back, so what is this? This is generally skin. I will enable my camera. Okay. So in most of people, so what is this? This is cell. This is skin.
Yeah.
If you open the skin, what happens? So this skin is a full length bodyguard of our body. It is a
Yeah.
security. It is a security. If you open, so there are some fats are there. The white
Oh,
color
yeah.
or yellow color fats will be there inside the skin. So what they allow, they will not allow easily the inner things to outside. So they are very strong. So in the lipids also, they are the fats. So they will not allow the things to leave, and they will not allow to leave easy. They will not easily digest. That is the process. Okay. If you take fully oil food, you will get a digestion problem because it can't digest easily. So in those kind of situations also, so this kind of archaea will live. So next, see here, what is the word we are getting? Permeable and semi-permeable. Okay. Permeable means which allow easily. Everything is come inside, can go outside. Semi-permeable means only certain things get inside and certain things get outside. Okay. So lipids that compose the cell membranes of archaea and are different from the lipids in eukaryotic cell membranes. The cell membranes of archaea form a unique structure that remains fluid and selectively permeable over the wide range of temperatures. So it is not easy digestion. Okay. So you need a lot of energy to digest the fats. It is not easy. Okay. The fiber can digest easily. So normally in the wheat, when you see the wheat, so you can see the outer layer as a fiber. So that can easily digest, easily can break up easily. But these lipids cannot break easily. We need a full energy and a wide range of temperatures we need to digest those things. Okay. And now different between bacteria and archaea. So despite their name, bacteria are not most ancient groups of organisms. So bacteria is the first one. Then later the archaea has been formed. The DNA studies have shown that bacteria are the most ancient group and that the archaea evolved from the eukaryotic cells in the latter time. Okay. So in the beginning, only the bacteria has been formed, but then the latter billions of years later, so these bacteria evolved to archaea, and which is from eukaryotic cells at a later stage. The cells of bacteria and archaea are different in several ways. Can you read up the first one?
For type of lipid structure in the cell membrane.
Okay. So there are different types of lipid structure in the cell membrane. The cell wall in bacteria contains peptidoglycan, but the cell wall in archaea does not, although there is a small compound of in some archaea. Both have different metabolic system, but methanogenesis, in which methane is produced, in unique to archaea. So, what is this archaea? How the petroleum reserves will form in the deep earth. In the Middle East, the oil they squeeze the oil from the earth, from the deep earth. How there the oil has been formed? Do you know? How can we produce oil, petroleum, other gases?
But
Where it can be
fossil fuels?
from fossils, from fossils. In the billions of years back, due to some asteroid hit to the earth, so all animals were died. So their bodies were collected one place, and they left like that only. After years and years and years and years, they covered with some dust and they went to the deep of the earth. But in those bodies, the skin is fat. Do you know the skin is fat? The skin is fat. It is not easily the things go to inside the things go to outside. So the remains of animals of their skins, each and everything collected together, and after the years and years, they released methane gas. So that's why nowadays the oil extractions, it is not oil extractions, it is a methane gas extraction. So it is a billions of years it will take time to become the methanogenesis. Okay, so that is in archaea is only form. Okay? Archaea is just very solid thing. It is not easy to uh, digest or easy to exist in the system. Okay, next, eukaryotes. Okay, so let's continue this in the next class.
Yeah.
Okay, so it is 7/5/25 at the rate 4:35. Okay, so I will recall each and everything in the next class about this bacteria and archaea, which are prokaryotic. Okay, in the next I will go a little bit advanced. If you see these eukaryotics are unicellular also here. So you can see animal and plant cells. They are the main two types of the eukaryotes. Okay. So
Okay.
it is basic understanding. It is a general topic. Okay, I already covered this. And see what is this for the first one? It is a bacteria, archaea. Okay, and it is cyanobacteria, and this is fungi. Okay, it is a fungus. You can see it is a fungus. It is a protist, and it is a plant cell. It is an animal cell. Okay, in the plant cell, chloroplast will form. So due to this chloroplast, the plant cell looks like always green due to this chloroplast and chloroplasma. So it is helpful to the photosynthesis process, we can see. And here it is vacuole. In the plant cell, vacuoles always with a big size. But in the animal cell, vacuoles are in the small size. Okay, this is the most important difference between these two. Okay, so I will stop here.
Yeah.
So in the next class, we go with rest portion of the chapter 2, and right now here I would like to stop now. Okay, so for the live students, it is botany, and we'll continue in the next class. Thank you, all the best for the next session. And evening, I think I had a mathematics class is there as usual at 7 o'clock or 7:30. Bye. So